Well, once again, hello and welcome to today's webinar, the Phytobiomes Alliance webinar. My name is Dusty Gallagher. I am the Executive Director of the Phytobiomes Alliance and Project Manager of the U.S. Culture Collection Network. So before we get to our speaker today, I would like to just spend a few minutes talking about the International Alliance for Phytobiomes Research. We are an international consortium of industry, academic, and governmental scientists. We are nonprofit, and our vision is that we can create an environment where all farmers have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions. And this will allow them to determine the best combination of plants, management practices, and inputs for a specific field in a given year. We couldn't do what we do without the generous sponsorships of the following companies and academic partners, research institutions. So we just want to take a minute to acknowledge them for their support. So once again, we also always like to talk about what a phytobiomes is. And so we know that phyto means plants, biomes means a specific site or substrate. So for this particular image, we're talking about soil. And then it encompasses all of the complex systems and interactions that influence those the plants in that substrate. So today we're really going to be focused on microbes, ones that live and interact in the soil, in the plants, in other animals, and that we know that the phytobiomes is also impacted by climate, weather, water, and then it's also influenced by management practices. So when we talk about the phytobiomes, we're talking about this complex system of plant-based agriculture. And so the microbes part of the interaction is what we're going to talk about today. And so the Alliance has a project, which is the U.S. Culture Collection Network. And that is an NSF-funded research coordination network that brings together scientists working with living microbial collections. And so USCCN is a partner with the Phytobiomes Alliance and NSF for that funding. And a major focus of the USCCN is that we've been developing a public registry and database, and we're focused on maximizing synergy among the collections that exist and the researchers who use them and anyone that's interested. And this is a collection that is a collection of microbes that is represents all types. So this includes the small research collections at universities, government agencies, large collections at universities, private for-profit and nonprofit organizations. So we are open, this online registry is open to all those collections, and the information is voluntary, but this is very important in the work that we do to help provide a sustainable future for these very important resources that we're going to be talking about today. And I would just like to mention that USCCN will be an exhibitor at the ASM Microbe Conference that's coming up later this month in Los Angeles. So if you happen to be in attendance, please visit us at booth 830, and we'll also have a poster on display on the Friday of that conference. And so webinars are a very important part of what we do at the Alliance. And so I just want to give you a heads up about the next one, which will be September 10th. And we will, with that webinar, we will be discussing designing biosensors to inform ecosystem engineering. And we'll have a presenter from the Oak Ridge National Laboratory. So if you haven't already done so, the registration is live. Please register to attend our next webinar. And so a few housekeeping items before we begin. This webinar is being recorded and will be posted on both YouTube channels of the Phytobiomes Alliance and the USCCN. And following this presentation today by our speaker, we will have what I hope will be a very robust Q&A session. So in order to interact and submit those questions to the speaker, we ask you to please use the questions panel. The chat panel is there, and you will be receiving information from the organizers. So that will be our one-way communication tool to you throughout the webinar. And then you will also see the handouts that will be available based on today's discussion. So it is an honor to be able to have today and introduce a partner that's been with USCCN for a very long time and has been around and providing a valuable service for a very long time, and that is the ATCC. And so we're very honored today to have Victoria Knight-Connoni, who is the head of content and product development at ATCC, and she's going to talk about 100 years of collecting biological materials. So without further ado, I will stop sharing my screen and invite Victoria to the stage to present her presentation. And welcome, Victoria. Thank you for being here with us today for this presentation.
Great, thank you. Thank you for the introduction, Dusty. And it really is an honor to share, what we have been doing at ATCC. So a lot of this is going to be telling you about what we have in our collection, but hopefully this will generate a number of questions. Okay, so I'll start with an introduction about ATCC in general. Then I'll tell you about some of the collection that we have and some of the products that we have been developing from that. I'll talk about some of the challenges and opportunities that we face at ATCC, but I am sure that a number of other people who have culture collections are also facing these challenges. And then I'll talk about some of the services we offer and then end with a summary. So just by introduction, I joined ATCC about 3.5 years ago, and at this point really was to oversee the collection that we have, to bring in new things. So we're always looking to make sure we're updating our collection with new items, to make sure we really have the resources there for the scientific community. That is our job, is to make sure we have materials, but also to highlight some of the things we have in our collection. As I go through this, you're going to see the collection is big, it's really diverse, but there's a lot of things that are not as well used as I think they probably could be. And then I also do oversee some product development where we take some of the materials we have in our collection and make derivatives from them that can be used also in research. So again, a little bit about ATCC. So we are really excited to be celebrating 100 years of existence this year. We just had a big gala to celebrate that event, which was a really nice way to bring employees together and really, sort of celebrate the successes that we've had. We are private and we are non-for-profit. We do not receive any funding from the government. The collection, it supports itself. So we do charge for our materials, and that really is to keep the collection going. At this point, we have a large collection of both cell lines, microorganisms, and then some derivatives that we have made from those. We do produce all of these materials under ISO standards, so we are trying to produce quality materials that each time you come back to us for a material, that is going to be the same as what you purchased the last time. I'll talk about this a little bit later, but we are really conscious about minimizing the number of transfers that we're making from our products as we put them into our catalog. So this is a busy slide, but I'll lead you through it. So again, we were founded in 1925 when there was sort of need that we should probably have a place for the scientific community to be able to access materials. And then a couple of years later, for many, maybe for some of you on the line, you may remember that it used to be an ATCC catalog that sat in people's labs where you could look through and see what is in this collection. This has obviously been since replaced by a website where we have all of these materials. Obviously, in the 1930s, our job was made a little easier and that we were able to freeze the materials and lyophilize, which made it much easier to be able to share materials out. So then the late 1950s, the NCI proposed having a collection of cell lines, and we were designated as a place to store those. We also, in the late 1970s, there was the Budapest Treaty that was signed, which meant that if you were depositing or publishing a patent, you needed to store that material in a collection, but it only needed to be in one collection. We had been storing patent deposits for people way before that, but this was when we started doing it under the Budapest Treaty. So in the late 90s or early 90s, we got a new CEO who really transformed the company and made it what it is today, and allowed us to become able to support ourselves. We have along the way gotten a number of contracts from the government sort of to support their materials. We have one for a repository. We also now have, I'll talk about our sites later, but we do have a manufacturing site in Manassas, Virginia. So it's allowed us to be in one larger location and expand. Again, we have gotten a number of government contracts. We do have the ATCC collection, which is the one I will spend my time on today, but we also, as part of the organization, do have the BEI collection as well. This is under contract, and it is the collection from NIAID. Some of those items are the same, but many of them are unique to each of the collections. We do also have a contract for influenza items through IRR. And sort of since starting, we have expanded into different areas. Again, in the around 2010, we started doing product development in a major way. So taking some of the items we have in the collection and making derivatives on them that could be used by the scientific community. Again, obviously during COVID, we were able to support some of these efforts by having new isolates available both within BEI but also within the ATCC collection. And then we have recently gotten an award to actually build out a new biomanufacturing site at our manufacturing headquarters in Manassas. And then with the transition to 100 years, we also have just recently appointed a new CEO from within ATCC, and she is likely, is going to take us to the next level. So again, we have 4 sites, the main one being in Manassas, Virginia. This is where all of the production of all of our cultures are done. This is where they are all stored. We have also a site in Gaithersburg. This is the site where I work. It's where our product development team is. We also do have a small repository there and a repository under CGMP conditions. We have a scale-up plant in Germantown. So this is sort of the bridge between Gaithersburg, our product development site, and Manassas, our production site, as we want to scale up new products or new deposits coming in. And then we also do have a small site in Frederick, Maryland, which is under a contract through the NCI. This is a repository for some higher level items there as well. So, ATCC, we are really proud of a lot of the reference materials that we have. These reference materials can be used in a number of different research areas. And, we were the first resource organization, so culture collection, to be accredited by ANSI. And we have a number of different standards, ANSI standards sort of around characterization of our organisms. So one around anthrax, a couple around characterization of our cell lines by STR and cytochrome C oxidase testing. So we also, ATCC is this nonprofit. We sort of work with a number of different organizations. I already mentioned some of the federal contracts that we have. So BEI being the other culture collection, IRR and NCI, but we also have another number of collaborations with industry and academic groups. So I'll touch on this later, but we have a collaboration with One Codex. This is the group that supports all of the genomes that we have. So the genomes for all of our organisms are stored on our site. They are not stored under NCBI, and I'll touch on that later. We have a collaboration with USP. We recently released a set of DNA products from cell lines. These are many of the commonly used bioproduction cell lines that could be used for host cell DNA detection. We have a collaboration with QIAGEN. This is around transcriptomics and and whole exome sequencing, of a number of our cell lines. These are available through QIAGEN, and there are about 600 of our cell lines available in that data set today. We also have a collaboration with HCMI. This is where we have a number of organoid models. These are patient-derived and come with both metadata, but also sequences. We have a collaboration with InSphero. This is around a set of spheroid products that can be purchased. And then we also have a collaboration with Edico, where they have a number of our cell lines where they can rapidly modify them using CRISPR. So it makes it very easy to be able to work with them to modify some of the cell lines we have in the collection. So although most of the products that we sell, we distribute ourselves within the US, we are also able to distribute these worldwide through a number of different distributors, so shown in the green, and then some international direct shipments to some of these countries. So it means our reach is far broader than just the US. It goes across the world. So a little bit about our collection. So again, the collection is big and it is continuing to grow. Again, I am a microbiologist by training. I have spent all of my career at this point, prior to coming to ATCC and industry, using microbes really as the starting point for products, both in sort of the biopharma space and under antibiotic production, but also more recently in agriculture, in using microbes that could be used to provide solutions to both abiotic and biotic control and biot stresses. So I will say it's a big collection. One of the first things I did when I joined the company was took a look at what was there. And I'd isolated microbes from a lot of different places, but I was really surprised by the diversity of this collection. Again, I thought I knew a lot of microbes and a lot of fungi, but there are things here for which I just did not know. Which again, as a microbiologist, this is sort of exciting. There's some really interesting things here, and obviously I think that there's a lot more potential to this collection than perhaps is being realized at this time. So again, we have a large collection of microbes. Both bacteria and fungi make up a good proportion of this collection. We also do have type strains, and then we have a number of different human and animal cell lines. And then, as I mentioned before, we have a number of derivatives. These are things that we've made from our collection. Again, we are proud of the fact that a lot of our strains are referenced by a number of regulatory agencies. So there's a number that are referenced in USP chapters, ISO, FDA, etc. We also have a number of strains that are used in quality control. And when you see this, if you download the slides, you can see there are QR codes, which will bring you very quickly to each of these sets of products. So this is a different slide than I normally talk about when I talk about our collection. But, why do we care about having so many things in the collection? And it really is that you never know what you're going to need. The example I normally give is about Zika, but I wanted to change it up this time. So again, earlier, sort of last year, we were talking, there was a report from a French group that said sort of Camembert and Brie were facing a threat because there was a decline in the microbial diversity they were using to produce cheese. And this was partly the strains they were using had been selected for a very specific purpose, but it was then decreasing the diversity, which was actually impacting what they could, the types of strains and the cheese they were producing. So again, this is when we looked at, well, what do we have in our collection? And we actually realized we had a number of different strains of Penicillium cambertii that were isolated from various different cheeses between 1904 and 1984. So again, every time we see something coming up, we could go back into the collection and say, well, what do we have that could be used again by the scientific community to actually be used in research. And again, there's a link here to a blog by one of my team members that will talk about this in a little bit more detail. So again, I'm not going to spend a lot of time on the cell biology part of our collection, but there are a number of really important products there. So we have more than 4,000 different cell biology products from a large number of species. We have both primary cells, so cells coming directly from tissue, but we also have a number of immortalized cells as well. As mentioned before, through HCMI, we have a number of these patient-derived cancer models, and they come with both clinical metadata and sequencing data to support those. And this will actually be coming out in a publication with those collaborators this year. We have a number of cancer cells with profiles, and then, as I mentioned, we do have DNA available for many of these. Just like for our microbes, as these come into the collection, they do get authenticated. We want to make sure that we are providing materials that are clean. So these go through STR testing or cytochrome oxidase testing to prove they are what they are supposed to be. We obviously want to make sure that we are not introducing contaminants to anybody's lab. So these are all tested for mycoplasma and for sterility. So again, we want to make sure that these are all good, and this happens at each production lot that we do. So then onto the bacteriology collection. So again, this is for the bacteria. Again, there's more than 1,200 different genera. This is not, of course, getting to the number of species. That is far more than that. Again, as a natural product microbiologist, I'm really excited about the number of streptomycetes that we have, but we also have a number of important genera there as well. And again, the diversity is what really, I think, sells this collection. You can see that these come from around the world and from more than 1,200 different institutions worldwide. So again, these also, when we bring them in, a depositor will provide the material to us. We will do QC and make sure, first of all, that it is pure. That is only one thing, and that it is what it's supposed to be. This is done, through marker sequences, so 16S ITS first, although all new products coming into our catalog immediately go for whole genome sequencing. This is done with both Oxford Nanopore long read scaffold and Illumina fill-in. And then we also will do some additional testing depending upon what the product is. So we will look at phenotype, but we will also do antimicrobial testing, MALDI-TOF to add to our library, and also serotype if needed. One of the things that sort of sets the collection apart is that when materials come in, we immediately make safety stocks. These are the stocks that almost never get touched. Then we make from those safety stocks a set of seed materials. These are the ones for which we make distribution materials. So for all of the products we have in the catalog, we have distribution materials that we are able to send out. These obviously do have to get replenished. And I'll talk about the challenge of that a little bit later. But again, the key to this collection is that we are monitoring the number of passages we get. So safety stocks almost never touched. The seed stocks are the ones we go back to so that we control the number of passages that these microbes, fungi, and cell lines actually have done. So obviously with the name like American Type Culture Collection, we have type strains. These are, as you people are describing new species of bacteria, fungi, viruses, they need to be deposited into 2 culture collections in 2 different countries. We have a number of these type strains. And again, these are always accepted into our collection without, if someone says they would like to deposit us, we always take these. This is different than our general deposits, which I'll talk about later. And again, these, as they come into the collection, although many times now have been sequenced, the genomes have been sequenced, we are sequencing them again so that we have the genome sequence associated with the microbe that is in our collection. And again, there are QR codes here, so you can see both what type strains we have, but also how to do this. We also have a set of strains that are part of a National Park Service special collection. Again, these ones are, if people are going out and getting permits from the Park Service, then the Park Service would like them to be deposited within ATCC. When people do this, you sign an agreement both with ATCC for deposit, but also with the National Park Service to make sure that they are getting recognition for materials that were isolated on National Park Service lands. These are some of the most interesting strains in our collection. Obviously from the picture here, we have strains that were isolated in hot springs. Obviously, the one that is the most well-known here is the strain of Thermus aquaticus. But there are lots of thermophiles, alkalophiles, acidophiles. But we also do have, from the Park Service, we have strains that were isolated from a cranberry bog in Massachusetts. So these are very diverse, but again, we want to make sure that we are preserving things from our national parks. We also have a set of strains that were isolated from the human microbiome and from the human microbiome collection. So again, they're everywhere you go, microbiomes are a buzz from people, but from plants also. But I think there's more and more knowledge about what microorganisms can do and how important they are in breaking down food, protecting us from disease, influencing our immune system. And so we have a number of strains that are part of this project. They are fully sequenced, and the metadata associated with these strains is fully documented. Again, you can access these materials from this QR code. Obviously, again, one of the things that are sort of near and dear to my heart is antimicrobial resistance. Again, this is a problem that has been rising and I think is becoming more and more of a challenge. So we actually went out to source. In this case, we went out and sourced a set of microbes that were antimicrobial resistant, but also came from humans from different sites. So these, you can see here, they are shown by the genus and species that they belong to. You can see that for many of them, we have microbes from different infections. And for these, we actually have the genomes available for these organisms. We have the full metadata associated with them, but also the antimicrobial resistance genes. And the phenotypic data associated with them as well. I will say we do not have intentionally, onto this next slide, microbes that are resistant to everything. So we obviously want to make sure that we are careful about what we are distributing. So we intentionally have picked strains that are sensitive to at least 2 different classes of antibiotics, so that obviously they can be treated. We did that in an intentional way. So we went through the collection and made sure that we do have strains that are sensitive. And so we do have this data available. I will say that this is being sort of added to the collection now. And actually, for those of you attending ASM, there is a poster on these this collection that will be presented there. And again, all of this information you can actually access on the certificate of analysis for all of these microbes. So also fungi, we have a really big collection of fungi. Again, more than 1,800 different genera, again, sourced around the world from a large number of institutions. Again, covering a lot of different phyla. Most of them are Basidiomycota or Ascomycota, but we do have them from many other phyla. Although you'll see we do have some gaps. So if anyone has fungi from any of those, we're always looking to add to the diversity. So we would obviously be interested in adding those to the collection. So again, one of the things that we've done with the fungal part of our collection, because of the metabolic creativity of this set of organisms, is we've actually looked at them and said, well, how could they be used in industrial processes? And I will say, for many of these, it is sort of guilt by association. So it is, okay, an organism of this genus and species has the ability to do this particular property. Ours may too. It has not always been confirmed, but it is at least a starting point for if you want an organism that is a plant pathogen. There are 8,000 of those that could potentially be used here. Again, so you can see the diversity of this set of organisms is incredible. And again, I would say this is one part of our collection that I think is not well utilized. I think there are many things that could be accessed here, and our hope is that we can find uses for them by the scientific community. So in the case of virology, again, a large number of organisms, viral strains here, sourced around the world. Again, large number of genera. Obviously, in the case of viruses, as with the microbiology part of our collection, we are always looking to make sure that we have new clinical isolates. So during COVID, we added materials here of all the newest versions of SARS-CoV-2. We have been sourcing the isolates of both monkeypox and measles that have recently come out. And in some cases, we can add those things directly to our collection. In some cases, we can only add the nucleic acid or an inactivated material. So obviously, in order to make these available to as many people as possible, if it is a BSL-3 organism, we will offer an inactivated or nucleic acid version of it, so we can get a BSL-1 product that can be used. Again, lots of interesting organisms in this part of the collection. So what are we doing with these? Again, we have a big collection, but as I mentioned, we want to make these available so that we are modifying these in many ways. So, we are taking our cell lines, our microbes, and developing products from them. So in the case of oncology, we are creating reporter strains. We released, actually last year, but also the beginning of this year, a set of CAR-T reporter strains that can be used. We also have a number of CRISPR-edited cell lines. In toxicology, we have both primary cells. We actually have just released a set of hepatocyte products, but then we also do take a number of our primary cells and create HTERT versions of them, so immortalized versions of them, so they can be cultured over and over again. We have a number of materials that can be used in molecular diagnostics. So these are, they can be live microbes, they can be nucleic acid, they can be inactivated materials. In the case of bioproduction, we have a number of bioproduction cell lines available, but we also have all of the materials that can be used for testing in bioproduction. I'll talk about that in the next slide. And then obviously data-driven solutions. So we are making data available for as many of our products as we can. And this is an area that where we are intending to grow. So I'll talk about that later. So in the case of bioproduction, obviously as people are producing products from both cells or microbes, you obviously need the production lines. We have a number of those. But then as you're also doing the production of these materials, then you need to do in-process testing. So we have sort of both sort of host cell, we have DNA isolated from a number of the production cell lines, which can be used as controls. We have a mycoplasma detection kit. Obviously, mycoplasma is something people are really concerned about. We also do have certified reference material, both DNA and microbe itself, available for mycoplasma. This is in support of microbes within the USP chapters. And then we do have also an Ad5 reference material. Again, all of these things can be used as people are producing products that can be used. And obviously, there is really a drive here to do this faster. So again, having nucleic acids makes this easier, but the time from starting material to end, driving that down and using molecular methods is obviously really important. We have just released a new set of products, and hopefully this video will work. So this is called MicroQuant. These are a number of strains that are in the USP chapters, and if you can see that they're going to add the diluent to the pellet. This is the microbe in this vial. You can see it immediately goes into solution, but again, we have already released the microbes that are described in chapters, the USP chapters 51 and 61. We will release this month, actually, the microbes in USP 60. And then later this year, the microbes that are described in USP chapters 62 and 71. So again, these are meant to make these products immediately useful by the scientific community. They are at low concentration and a higher concentration, so they can immediately go into tests. So then thinking about molecular diagnostics, again, this has become really important. Obviously, during COVID, everyone was doing tests. So molecular methods are really important for getting results very quickly. So as you think about the molecular diagnostic sort of assay development, you can see that you need different materials as you are developing these assays. So during, early assay development, you may need different materials than when you actually have your test finally complete and you just need routine run controls. Obviously, we need to make sure that your assay is sensitive and it is specific. And again, different organisms needed at different points. We have a number of organisms that can be used. So again, molecular standards, which I'll talk about in the next slide, but we also have live cultures and then some synthetics and inactivated materials. Again, these allow more wide access of the materials. Again, having these different formats really does allow rapid assay development. So again, with some of the molecular standards we have a number of different genomic standards. So organisms in our collection where we have the genomic DNA at a set concentration that is ready for use, that you can come back and get over and over again. But we also have a number of synthetic standards. Again, these ones are ones that we have designed. They are pieces of the genome of each of these different organisms. And the reason for these, they are oftentimes organisms that we are not able to access because we can't source them, or that they are of a BSL-3 level so that even if we could access them, most of people couldn't, and so they wouldn't really be useful. One of the ones that we have recently released in this space is in avian influenza. And so obviously this is one that people wanted to rapidly get, was not available for most people. So we have a synthetic avian influenza standard that can be used, and we have validated that it can be used in detection assays. Again, we also have inactivated SARS-CoV-2 and also RNA available so in this sort of suite of products that we have. We also have, and they're called sort of next generation sequencing standards. They were previously called microbiome standards. These are materials that are mixtures of organisms that we have both whole cell and nucleic acid mixes of these. They are both, you can have even distribution of the organisms in them, and sometimes there are staggered concentrations. Again, they range from 3 to 20 different organisms in these sets. And we have, on the microbiome, we have sets that come, come from the oral, skin, gut, vaginal samples that can be used. We do have a set also that are environmental. These ones are listed sort of under sort of a diversity set. Again, the information on these you can find with this QR code, but again, are very useful for sequencing. And again, when we are producing these, we are assembling these individually and then mixing them at the end to make sure that the concentration of those organisms is always the same. So I talked before about the genome portal that we have. So one of the challenges that we face as a culture collection is that a lot of people will order our microbes and they bring them into their lab, and then they may pass it to another lab down the hall. Again, I am equally as guilty of that when I was working in research during graduate school. But the challenge to that being is that there is, those organisms get passaged a number of times, and making sure that that organism is the same one is often really challenging. So the organisms in our genome portal are limited in the number of passages. They are the organism that you can order from us. We have about 5,000 genomes now. They are Oxford nanopore scaffold with Illumina fill-in. We are continually adding to these. The goal is to get everything in the collection, but you can imagine that's going to take us some time. But you can always tell us if you want something, if you're looking for a microbe, please let us know. If you order a microbe from us, you can always get that through the genome portal using the lot number. We do, though, for everything else, there is supporting membership to access these now. They are different prices to do that for academia and industry. And again, this is really meant to just help defray the cost of this. Again, our collection needs to support itself. We don't get funding to do this. So I'm going to go through this quickly because I realize I'm running out of time. So some of the challenges that we have with a large collection that's been around for a long time is the metadata. We know this, and we are working through trying to get control vocabulary. So we are able to mine the data better. So, for example, they may be listed as this was grown aerobically versus 100% air. Those are probably mean the same thing, but it's really difficult to mine these. Also media. Again, it's really challenging if, for the microbes, there are 1,200 different genera, but they may say they need to be grown in 1,400 different media. That's a lot of media to have available. And then, so can we actually get to a smaller number of media here? And then also the way, because there are so many things in our collection, and they grow under so many different conditions, it means we actually need a very skilled workforce to be able to produce these. Growing E. coli, very easy to do. But if you have something that grows high pressure under, very, anaerobic conditions, that isn't going to be as easy to do. So this is one of the challenges that we have. The other is keeping all of the products in the catalog. Again, I said it was a really big collection, but a lot of them don't distribute. So what do we do about that? How do we make sure we have these things available when people need them? This is why you sometimes see things in a collection saying it's a mission item. It's because it hasn't been requested a lot, and we will need to produce that if you actually want it. So again, this is one of the challenges we face. And then obviously, how do we actually distribute these worldwide? BSL-3 levels limit us a lot of times. Also making sure that we have permission for the Nagoya Protocol. No, the US is not a signatory of that, but we want to make sure we will are following it as much as we can. And then also for our cell lines in particular, the source of the animal byproducts is really challenging. So quickly, services. If you want to deposit with us, there's a slide here on how you can do that. We are always looking for new things. We get that both from people reaching out to us, but we will also source things ourselves and say, please, can we have this? This is in our collection. We store it, so it's always there. But we also, if someone takes a license with us, we do provide royalty back to the depositor. We also do have a service for CGMP cell repository. These are produced and stored under CGMP conditions and stored there for use. And then finally, we do have a licensing group. This is something I didn't know before coming to ATCC, but you don't just have to use our materials for research. If you want to use it for in a product, you can do that. You just would need a license from ATCC. We have a group that does that. But we also have a patent depository, which is different than our own collection, but this is where we store items that are described in patents. So finally, again, ATCC, I hope I've shown you, we have a very large collection. Really, the driver for this is that they are high quality, they are authenticated. You can come back and get the same product over and over again. We have a large number of partnerships with industry, government. But really, again, the message is we're here for you. What is it that you need? If there is something that you say, ATC doesn't have this, could you get it for me? We are always interested in trying to add these to the collection for the scientific community. Or if you need a different format, again, please reach out. So with that, I'm happy to take any questions. And again, we will be at ASM. I've put the booth number here.
Thank you, Victoria. That was a great overview of ATCC, and it's great to have all of those updates and hear about all the wonderful things that ATCC is doing. So we do have a number of questions coming in. And so I'm going to begin with something that you mentioned in one of your last slides, and that was about the Nagoya Protocol. And so we have a question here that maybe you can elaborate on. It says, the question is, even though the USA didn't sign the Nagoya Protocol, how do you manage the deposit requests coming from signatory countries?
It's complex. I mean, we do the best that we can. So again, we have a legal group that sort of looks through those to try and make sure that they do have the documents they need to show that they are collected correctly.
There's I guess another agreement, the new Biodiversity Beyond National Jurisdiction Agreement under the United Nations Convention and the Law of the Sea. Are you familiar with this? And does ATCC have any plans to comply with this?
So I will say no, I am not, but I will certainly look into that.
Okay, all right, great. Next question is about your pricing. Do you have a specific pricing policy and is that published on your online catalog, or does it differ just depending on what strains that a customer may be interested in?
So we have pricing that differs. The biggest difference is there is pricing for nonprofit and government, so academic and government agencies, and then one for industry. Those are different. It usually depends on what the organism is. So if it is just a microbe, it would have a price. If it is been modified in some way, then it would be a higher price. But there are also, I mean, there are some organisms, that are at lower prices because they are used very regularly, and so it's really about. So we'll say we have a whole product management team that reviews all of that and is trying to make the best prices for everybody. And again, I mean, yes, it does. We do charge for them, but it really is that the organisms that sell are paying for the collection. It's to keep us in business.
Mhm. Which is very important. Okay, our next question is, what are the conditions for using the strains after distribution indicated in your MTA? Are there any restrictions to commercial use, or do you have separate agreements for, say, research use versus commercial use?
So the original agreement that is signed is for research use and research use only. But if people, and a lot of people do use the collection for commercial use, then there again, you just have to reach out to our licensing group, and then they will negotiate a license with the customer. And again, this is where I was talking about if people deposit materials with us, we will share that licensing revenue back um, with the person that deposited the material. So yes, this was the thing that actually I did not know before coming to ATCC. I had always heard that you can only use it for research use, but that's actually not true. It can be used in both places.
Okay.
Just there's a separate agreement that has to be signed prior to commercial use.
Mhm. Would you be able to share the staffing levels that support the fungal portion of your collection?
It's small. I mean, I would say it's less than 10 people. So there is a part of my group that is, so I have 2 people within my group who are looking at bringing new deposits in. So they are looking at characterizing those strains and looking for them. I'm reviewing them. In the bioproduction part, it's a small group. I think less than 10 is probably right.
Okay. And I'm guessing that they have very specific skills
They do.
in terms to be able to do that and do that
Yes.
the right way, correct?
Yes. Yes. I mean, I will say we are not I mean, as much as I said the challenge is keeping everything in the collection is a challenge. We do not produce everything every year. So again, we make distribution stocks, and the goal is to make stocks that will allow for having materials to be sold for a number of different years. So again, there's a there's a whole group that does the scheduling of them to try and make sure that we have materials available. But we often get caught off guard because all of a sudden something will come up in the news and people will want it, and then we're like,
Yeah.
we weren't planning on that many people wanting that thing.
Uh-huh. I imagine
But
that is a big challenge in itself to create that production cycle and
Yes,
production
absolutely.
calendar.
Yep.
Yes.
Yep.
Okay. Our next question is, what is your approach to biosecurity when it comes to screening new deposits? Do you screen both the depositor and whatever is being deposited?
Yes. Yeah. So actually when things come in, we have a list, a denied parties list, and this is actually both on who we will not sell to and who we will not receive materials from. But also the other thing that happens when someone says, I'd like to deposit with you, we will look at it first for its scientific merit. We no longer can accept everything um, because it's too hard to keep everything in the collection. So we will look at it and say, okay, do we think that this is going to be useful for the scientific community? But we also go through and say, does that person have the right to deposit it? Do they have ownership of that material and everything in that material? So if it had a plasmid inside, do they have ownership of that plasmid? Can they deposit with us? So all of that is considered. So again, we meet actually every week or every other week, depending on every other week for different parts of the collection. And it's a cross-functional group, and we are looking at all of that, both the scientific merit, but the legal part of it as well, and the regulatory. Yeah.
So you've touched on this in the previous question, but when you create stocks for distribution, how do you estimate what you're going to need to distribute so you can manage your freezer space? Is it strictly based on demand at that time, or how do you, what's the
So
strategy behind that?
yeah, so when new things come in, we will make a smaller batch of it to start with because we're not sure how it's going to do. Unless it's something that is we expect when we bring it in to be very high demand. So, for example, for the materials we brought in for SARS-CoV-2, those we expect to distribute really well. So we made a larger batch. But for if you're just adding, a new Pseudomonas strain, it would be a smaller batch first until we see that. But as far as, like I said, we have a whole group that's doing planning that is actually saying, okay, this is how this has distributed for the last few years. And so we can plan for that. So they say, okay, we'd like to have X number of years of this in our collection, ready for distribution. That's how they're doing it.
Mhm. Do you consider having a certain capacity, or do you feel like your capacity is unlimited based on how you manage what you produce for distribution?
We do not have it. So no, we do not have unlimited
Okay.
capacity.
Okay.
No, we are always looking to like that is something that we're always managing is to make sure that we, you know, I mean, that's obviously why you'll see in our catalog, you'll see things sometimes that are they're not available right now. And it's because
Mhm.
that we do run into backlogs.
Mhm.
Although some of that backlog is that, there may be a problem. We obviously don't hope for contamination, but sometimes, contamination will happen.
Mhm.
Or, you know, uh, you know, something may happen with the equipment. We may have a for cell lines, for example, they will not be released until they have been proved to be sterile. We have to go through a sterility test. And we did have a problem where one of the incubators we were using for the sterility testing went out of spec. And so we had to then retest all of that material, which when that's a 2-week test, that takes time.
Okay, well, we do have a few questions remaining that we will not be able to get to live, so we will work with Victoria to get those questions answered and get those posted to the website where you are all able to view them. And we just want to thank Victoria for this great presentation and discussion. And we want to thank all of our participants for joining today and look to participate in our next webinar in September. So thank you all for participation.
Okay, and thank you for the opportunity to tell you about the collection. Thanks.